Greetings, my name is Jorge Vilches. This video is part of my application for the REPU chemistry program. I'm an undergraduate, third year toxicology student from Universidad Nacional Mayor de San Marcos. My main research interest is drug discovery, and to be a good researcher in that area, my main objective is to be able to handle diseases that are important for me and that are very interesting for me, specifically in neurodegenerative diseases such as Alzheimer and Parkinson, by combining techniques from molecular biology and medicinal chemistry. Using the new techniques that we have already, such as bioinformatics techniques and chemoinformatics techniques, we are able to prepare and to know about new compounds that uh, no does don't exist yet. And I want my research job to be about around that, to be able to formulate new products and new molecules, and to be able to give people hope in a field I can contribute with, feel that I'm um, part of the change of these people's hope. My first research experience was in the molecular biology laboratory around January. And also my third research experience was around the same polymer, chitosan. The idea of the project was to formulate a specific concentration of chitosan that it's able to protect the alkaloid that has an antimicrobial effect. So the quinolicin alkaloids are very characteristic from the Lupinus genre. Specifically, Lupinus mutabilis is an endemic plant that has a very large amount of alkaloids. The idea behind this, it was as the alkaloid itself has a very strong in vitro antibacterial effect, antibiotic effect against fungi and viruses, and it has been already demonstrated. The decision behind the not using the alkaloid as an actual product is because of its poor physicochemical properties. This alkaloid is easily metabolized. It can cause a lot of secondary reactions and non-intentional reactions, adverse reaction from drugs, and also is cytotoxic, so it may kill or damage the patient. And the idea behind a nanoencapsulation of this alkaloid was to protect the person from the cytotoxic effects, to make the alkaloid specific so it can reach the actual places that are infected, and to take advantage of these characteristics to formulate a product. Chitosan is a very important polymer that my job based on this, my job was to prepare the formulations and the by freeze drying, by drying different concentrations of this chitosan and the combination with the alkaloids. And it had very strong challenges, such as repeating the same assays over and over again since it's a pharmaceutical product, so I have to make sure it's not contaminated and the sample is significant, has a great significance into my project. And the results I had been working on this laboratory were important to the conclusion of this project. As the best formulation was compared to the best formulation that has the best effect also, so we could decide which of them those formulation had the characteristics to be a good product. For my second research experience was in the same laboratory under the guidance of Dr. Arturo Intiquilla about uh, the comparison between ultrafiltration method as a new and novel method of purification proteins in the flower of Erythrina edulis. This was around this hypothesis that ultrafiltration not only has a very improved effectiveness over the extraction of proteins, specifically albumin, but also that ultrafiltration keeps the proteins safe from all the type of damages, such as unfolding the protein and losing the antioxidant effect that these protein fractions have. So, ultrafiltration method demonstrated that it could give all those characteristics, the antioxidant effects. It was more effective, even though it is not that cost effective. My results that were the calibration curve and the isoelectric point purification group as a comparison were very important to determine that ultrafiltration was the best one of them. For this experience, I had to prepare calibration curves using a BSA, bovine serum albumin, to prepare calibration curves for proteins. The Bradford reagent and combines with the amount of proteins that there is on the sample, and it can be measured. This correlation, this relationship can be measured by a spectrophotometry. My work was to prepare the curve of calibration and to measure the amount of proteins that it had in the isoelectric point purification, and also to measure the antioxidant effect of this fraction to demonstrate that this fraction, even though it sometimes could get a comparable amount of protein than ultrafiltration method, apparently, it didn't have the same activity because the isoelectric point purification harms structure of the functional structure of the protein. And it was very frustrating to deal with these type of purifications in a molecular biology laboratory. I was very new. I was generally crying when my supervisor told me that my results were great because I got an R square value of 0.95, and that was an actual decent curve that we could use to start measuring the proteins. I used a lot of samples, a repeated experiment over 2 weeks since I learned how to use the micropipette, how to use all the laboratory equipment. It was very frustrating, but at the end of the day, it was relevant. I felt that all the work that I was doing had a decent effect and conserved all the experience in this project. Even though it was not my area, talk me about also working with peers that are doing their jobs that are able to teach me about their methods and to be able to comprehend the impact of the things I was doing into all the researchers or the people around me and to all the work that we were doing in the lab. My final research experience was around something closer to my research objective. I participated into a project called formulation of an antiviral drug against chickenpox based on gallium and ferrite ions and quercetin. I participated in the formulation of the gallium quercetin complex by the synthesis of this complex and also the solubility test of these molecules. My results were important to determine and to characterize which of the solvents were more optimal for this specific compound. Also to determine to characterize the chitosan that was going to involve this molecule. The point of making a quercetin metal complex was to make the quercetin even more bioavailable. So we could try to use polyphenols such as quercetins due to its biological effects, such as antioxidant, antiviral, antibiotic, but it doesn't have a very good physicochemical characteristics to be a decent drug. So by by making a coordination complex, we are able to make the molecule more stable and to keep the biological activity. And by nanoencapsulating this molecule, we also prevented the make this formulation even more bioavailable, more specific, and more useful to treat the chickenpox in this project. My characterization of the chitosan helped the researcher to choose the best chitosan lot, the best chitosan to use, since we needed a low molecular weight chitosan to make the nano encapsulations in both cases. The samples I was taking and due to the experience I had into the laboratory to characterize these materials, my results were relevant to clarify my results were relevant to determine that the chitosan that we were using and we were about to use was medium weight molecular medium weight molecular weight chitosan instead of the low molecular weight chitosan. So we had to change the type of chitosan we were using, and I was able to feel that my results were actually being relevant. An important part of this and all these experiences was to learn the basics of the laboratory. It was challenging since it took a lot of time, but it also taught me about um, chemistry, basic chemistry and medicinal chemistry procedures. Finally, my extracurriculars are more around administration and being part of a community. National Research Direction, I am the national director of the research in this association of pharmacy students. It was very hard since it's about coordination to talking with people. I am an introvert myself, so I especially got this role because it involved challenging myself to do things that I was not expected to do. And I managed how to prepare the webinars to connect with the people from all the pharmacy schools and the student representatives from all these pharmacy schools around Peru. How to make them organize, to make good products and good and to produce good information for especially in the research part. I also part of the Future PCP, that is a club in artificial intelligence research. This is mostly composed of people from different backgrounds, such as engineering and law. So, being as the only biochemist in this group, I learned a lot of the perspectives that people had around artificial intelligence. Also participated into a webinar where we and other students around the country discussed about how AI was going to affect the future based on the MIT webinar that launched worldwide and talk about its content. It was an amazing experience, and it helped me to learn about everything about AI. As in CEFARP, that is the local student center from my school, my my faculty, I work into making the students know about the research groups in our faculty, the 22 research groups in our faculty, to make them know the lines, what do they do, why they are working on that, and believe that is important. And I personally feel that that was close and had a very important relevance and impact into the students that wanted to be researchers too. I also participated as a supporter in pharmacology and pharmacognosy teaching as a teaching assistant, and also in the pharmacognosy classes, I convinced my teacher and started to research about the potential metabolites of fungi, and especially Peruvian fungi, into pharmacognosy. The medicinal effects, the nutraceutical effects, and even all the characteristics that make fungi very attractive for pharmacognosy. I also took the English language teaching program, so it could be more connected with people that wanted to overcome this language barrier and to learn more about everything that amazed me about the world. Science, being able to connect with all these people, being able to be here and to talk with and to get opportunities and to know about these amazing people around the world. And well, this is basically my application. I am very grateful for being here. I'm very grateful for the person that will listen to this. I consider that I will overcome any challenge that comes in my way. Thanks for watching and see you later.